Today we'll go over what modulate the transcription factors and what makes them available or not in a gene expression. Gene expression is the amount of specific mRNA in a sample. There are 5 mechanisms that modulate the transcription factors, so let's begin. Transcription occurs in the nucleus, but the transcription factors is in the cytoplasm. Therefore, it's not possible to drive transcription from cytoplasm. They have to be translocated into nucleus to be effective and to carry out transcription.
Chemical modification, or the addition of compounds to transcription factors, occurs in the nucleus and influences the way those transcription factors work. An example of this is phosphorylation. Transcription factors have a specific binding site that allows it to bind to DNA. Phosphate kinases are the key molecules to phosphorylation. The addition of a phosphate group to a transcription factor changes the shape or conformation of the DNA binding site on that transcription factor. This inhibits the transcription factor from binding to the DNA and therefore influencing transcription.
Ligand binding plays another important role in gene expression regulation. When a receptor on the cell surface is not bound with a ligand, a pathway of activation and deactivation leads to no DNA translation within the cell. When a ligand does bind to a receptor, the activation-deactivation pathway leads to a switching on of translation in the DNA and the production of proteins within the cell.
Oligomerization of receptors also modulate gene expression. The bound receptor may need to be dimerized before transcription may commence. The androgen receptor, for example, is bound and translocated to the nucleus, where it is dimerized into a homodimer before it initiates transcription. So when the relevant configuration is met, be it mono, homo, or heterodimer, a co-activator can be recruited and transcription may occur.
An association inhibitor known as transcription factor inhibitors can modulate transcription factor function by directly and indirectly preventing them from binding with DNA. When foxhead protein FOXO forms a homodimer with FOX protein or heterodimer with an estrogen receptor, protein transcription is inhibited by way of oligomerization. FOXO transcription factors play vital roles in gene expression regulation involved in cell growth, proliferation, differentiation, and longevity. You can remember the 5 mechanisms as cute cats, like omelettes always.